So last time we talked about the process of DNA replication. We talked about in step 1, DNA helicase is going to break apart 2 strands by separating and breaking those hydrogen bonds between complementary base pairs. Step 2, you have DNA polymerase. It's going to come add base pairs. Now, that phosphodiester bond is added at this stage in step 2. We always move in the 5 to 3' direction. And if you don't remember what that means, remember the ribose, deoxyribose, the pentagon, the sugar in your DNA points towards the 5' end. So we have 5' on that end, which means that this is 3'. If this is 5 up here, that means this must be 3, and this must be 5, right? We call this anti-parallel. This means that DNA has kind of sides of the street, right? You always drive on the right side of the street. Well, DNA polymerase can only work in one direction. So if I do that again, here we have the 5 to 3, and the strand that already exists, we call the parent strand. The strand that's going to be formed, we call the daughter strand, right? And DNA polymerase can only work on the 5 to 3 direction of the parent strand, which means it's adding nucleotides in the 3 to 5 direction on the daughter strand. So there you go. We're going to add nucleotides in the 5 to 3 direction of the parent strand to form the daughter strand. Now, this is important on the lagging strand. This would be the lagging strand because you can see the polymerase has to work backwards. Now, that's a problem because it can only work in chunks, and then it has to wait for more of the parent strand to be split apart, and then it can start adding again in chunks, and we call those chunks Okazaki fragments. And lastly, we have DNA ligase, which comes through and it reforms those hydrogen bonds between the DNA strands. And that's kind of a summary of what we talked about last lecture. That was understandings 1, 2, and 3, I think, right? Maybe 4? I don't think so. Maybe just 3. And then today we're going to talk about A1. So 2.7A1, the use of Taq DNA polymerase to produce multiple copies of DNA rapidly by a reaction called polymerase chain reaction. Now, you might be thinking, well, why do we do this? So whenever you see humans manipulating DNA for an investigation, right? Or looking for DNA at a crime scene, or we're looking at specific parts of DNA that we want to study for genomic research, we need lots and lots and lots of copies of that DNA to experiment on and to examine. PCR, polymerase chain reaction, is a way that we can generate copies of that natural process very, very quickly. So we used our knowledge of DNA synthesis to devise a method using a particular enzyme to create DNA in a lab, create copies of DNA in a lab, much faster than would occur naturally. Think of it as like a DNA photocopier. So I'm going to show you a quick video that goes over the basic principle, and then we'll talk about it in detail.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Okay, so let's go through that step by step and talk about that process. In order to study genes and use them in experiments, we have to create a large amount of that particular piece of DNA. And the process by which we do that is called the polymerase chain reaction. Now, that makes sense, right? We use polymerase because poly means many. Ase is an enzyme, right? So we know that polymerase is an enzyme that creates polymers. Remember, DNA is a nucleic acid. It's the polymer, right? Nucleotides are the monomer. DNA polymerase creates DNA. Now, the reaction, polymerase chain reaction, was actually developed by a guy. He's a scientist out in California, and he was riding his motorcycle on the Pacific Coast Highway. And he was out for a drive on a weekend, and he was thinking about it, and he saw the sign that said, and the symbol for the Pacific Coast Highway is like PCH, or no, Pacific Coast Roadway. That's what it was. It was PCR. And he saw PCR and he thought polymerase chain reaction. And that's actually how he devised the method. So he was out cruising on his motorcycle, saw a sign, and a light bulb went over his head, and the rest is history. So not all discoveries are in the lab. What you're looking at here is a thermocycler. See how it says thermocycler? A thermocycler, you can think of it as like a tiny oven that you can set to different temperatures for different times. So here's the keypad, and you can say, okay, I want this to heat up to 95 degrees Celsius for 20 minutes, and then I want to bring it down to 15 degrees Celsius for 30 minutes. So it's kind of like an oven, but you can, program multiple cycles of heat. Thermocycler. These are really important in this process because if you noticed in the video, there were different temperatures at different stages. The first part, you had to heat it up to get the DNA to split instead of using helicase, right? And then during other processes, you have to bring that temperature down. And that's a good indication as to why this process doesn't happen at this kind of speed in the human body or in any animal for that matter, because our body temperature doesn't get that hot. And if it did, right, our molecules would denature. They would change shape. Okay, I'm getting all over the place, so let me go back. Okay, so in order for the polymerase chain reaction to occur, you have to have all the necessary components for DNA replication. So you put all of these things in the test tube. You have your sample DNA. You have primers. Now, we didn't talk about primers when we talked about DNA synthesis. All they are molecules that bind to DNA that tell the molecules where to start copying DNA. So you can think of it as like a start here button, right? And those primers fit to a certain sequence of base pairs. So they might only fit to a certain sequence that's CTTGA. They're going to look for that sequence and they're going to bind there. And then when that happens, that's going to tell DNA polymerase, okay, this is where you start adding copies. You need, of course, lots of nucleotides with all the different bases because these are your different pieces that you're going to string together. You need your Taq polymerase. Remember, that's our enzyme that's going to add those base pairs. You need mixing buffer, which is just the solution that all this happens in. And of course, you need the tube itself. So all these things go in the tube. And then you put the tube in the thermocycler. And you run it through. Now, DNA primers, this is kind of what I was just talking about. DNA primers are used to isolate the gene that you want. Now, in the video, we weren't making copies of the entire genome. We only wanted a certain piece of DNA because we're studying that piece, right? So you can design your primer to fit a certain length and a certain sequence of DNA that you find at the beginning of that gene and at the end. That way you're telling the Taq polymerase, I only want you to copy from here to here. It's like when you're copying somebody else's homework and they're like, only copy this part, don't copy the rest because your class didn't get assigned that, right? So you don't want to copy the whole thing. You just want the piece that you need. And again, remember, look, we're going from the 5 to 3 direction. So this would be your forward primer. This is going to tell Taq polymerase where to start. And this is going to tell it where to end. So now let's talk about temperature. So during the process of PCR, we're going to cycle those temperatures up and down. This should say denaturing, not deneutering. I don't know what deneutering is. Denaturing. Denaturing. And that's the same denaturing that we talked about like a million times already. When you heat something up to the point where you start breaking bonds in that molecule is going to change its shape. When it changes its shape, it can't do its job. So, remember, we have nucleotides that have hydrogen bonds in here. So when you heat up this DNA strand, that effectively breaks those hydrogen bonds. Now, we don't heat it up to the point where we start breaking the phosphodiester bonds on the outside, so we're not breaking apart the nucleotides. We're just breaking apart down the center because those hydrogen bonds are weak. They are weaker than the phosphodiester bonds, so they break first. So we're going to heat up to almost boiling, and that's going to cause the separation of the two strands. The next stage is called the annealing stage, and you might know the word annealing from blacksmithing, right? So when people that use forges to create items, metal items, so of course I think immediately like samurai sword. But in reality, right, anytime you're using an old style forge and you're sticking something in the fire and you're heating it up, right, so that it's nice and soft, and then the blacksmith pulls it out and he starts hammering on it and it cools down, that cooling down stage, that's the annealing stage. And just like with the blacksmith, this is the point at which you start building. So those primers attach to the DNA strand, and that primer tells Taq polymerase where to start adding the base pairs. So denaturing, we're going to heat up and split the strands. Annealing, we're going to cool down and let the primers attach. And that's about 50 to 56 degrees Celsius. The third stage is extending. This is when the Taq polymerase starts adding those nucleotides and extending the DNA strand. So this together is one cycle, and this will create one copy of the gene. Now you'll notice that I created 2 copies of the gene, right? In this one process, because remember, DNA is double-stranded. So this might be the coding strand of the DNA, and this might be the template strand. meaning the information of the gene is actually only in this one, right? Which means the gene I actually copied is right there. This is going to be another template strand, but that's okay because the next time around, now I have 2 template strands to make another copy. So the next time I make it, I'll have 2 more copies. So the extending stage, you're using a medium temperature that's going to add those nucleotides, and it's DNA polymerase that's allowing that. So let's look at that again. Oh, we'll look at the video again at the end. So this particular enzyme, Taq DNA polymerase, it's isolated from a species of bacteria, T. aquaticus, and it's found in hot springs. So its enzymes are designed to work at high temperatures. Remember, we talked about how different proteins have different optimal ranges of tolerance, right? They function at different temperatures. Some species live at very warm conditions. This is a throwback, right? Some work under very hot conditions. Some work under very cold conditions. We talked about how different enzymes work in your stomach versus your intestines. Your stomach is very acidic. Your intestines are very basic. So we specifically looked for a DNA polymerase in a species that could exist at very high temperatures because we knew that using temperatures was going to be important in this process. We found that polymerase in the species of T. aquaticus, right, that normally functions at high temperatures. And this picture is from Yellowstone. I took that picture. It's from the thumb paint pots in Yellowstone Park. Okay, so each cycle of PCR doubles the copies of the target gene. So you produce a large number in a short period of time. So one cycle means denaturing, annealing, and extending. That's one cycle. So one cycle gave us 4 copies. Now you might be saying, why is that 4 copies? Because it's only 2 strands. Remember, DNA has complementary base pairing. So the gene, or in DNA in general, you have 2 strands. You have the top strand, which is called the template strand. And this will make more sense when we talk about transcription and translation. The bottom strand we call the coding strand. So the coding strand actually holds the nucleotides that tell the mRNA what codons are and what amino acids to put together. But the template strand, because of complementary base pairing, right, it also contains that same information because it's complementary. So A, T, right, G, C. So if I make the complementary strand to this again and again and again, I get more of the gene. That'll make more sense later once we talk about transcription and translation. For now, just understand that you're going to get, if you start with a double strand, right, you're going to make 1 copy of that double strand, which is then 4 copies of the gene. So now we take these 2 and we do the process again. And then we do the process again. The picture is cut off. Actually, this should be this one, and then there should be 2 more down here. The picture got cut off, and you can see how this goes. So every time you do the cycle, you're doubling. It's an exponential increase, right? Just like bacteria dividing. So we can make lots and lots of copies in a pretty short period of time. Okay, so at this point, you should be able to discuss why DNA replication is important for organisms, right? You should be able to outline the role of helicase and DNA polymerase in replication. You should understand what semi-conservative means when we're talking about DNA replication. You should be able to analyze the findings of the Michelson-Stahl experiment, that was last rotation. And you should be able to outline the use of Taq polymerase in PCR, including each step in one cycle of the process. Okay? All right, so I'm going to pause the video or chop this video off, but that doesn't mean you're done for the day. Make sure you continue on to transcription for today, okay? So make sure you upload your notes and continue on to transcription, and I'll see you in a few minutes.